Hi guys. So really quickly, we are just going to go through cells in general. So after your notes that you've taken on 7.1, after the cell mobile activity that you did on your personal organelle, and then after our activity where we walked around the room and looked at other people's organelle mobiles, I just want to make sure we're all on the same page and that you have the information for the cell unit thus far. Okay, so what I want you to do is I want you to watch this video and take notes as we're going along. Fill in your notes with everything that you might have missed, and also make sure you are using that table that you made in class that has your name of the organelle, where you sketch out its structure, where you list its function, and whether you say it's in a plant or an animal cell. Okay, so this is just kind of like a wrap up. Make sure we all have the information all in one place. So the purpose of this little video is so you can know the scientists who contributed to the cell theory. Remember, that was our timeline activity. List the components of the cell theory, compare prokaryotes and eukaryotes, label a plant and animal cell, and know those function of those cell organelles. So remember, the big players in cells and cells theory was Hooke. He was the first person to actually see cells. And Leeuwenhoek, he looked at cells in pond water, but he actually called them animalcules. So this is what he saw, his animalcules. Now, there are three main guys that I want you to think about when you have the cell theory: Schwann, Schleiden, and Virchow. Schwann was the tissues of animal cells. Schleiden was the botanist, which means the plant cells. That's when he discovered, oh, plants have cells as well. And then Virchow, he also reported that every living thing is made up of vital units known as cells. He also predicted that cells come from other cells. Remember, guys, it's really important to know that how all these discoveries were made and how they were able to build off of one another is because microscopes not only were one invented, but then they got better and better and better. So that helped us to see the cells even more, and these early scientists to investigate them. So you should know the three parts of the cell theory. One, organisms are made up of one or more cells. The cell is the basic unit of structure and function, or the basic unit of all life, and all cells come from other cells. Those are the three main points. So remember, in your reading and your first set of notes that you did, I think that was last Wednesday, e-learning day, you learned about the two types of cells, and prokaryotes and eukaryotes, and there are things that make them different. However, all cells have these parts. They all have ribosomes, which make proteins. They all have cytoplasm, which is that jelly-like fluid within the cell. They all have DNA, that's the genetic material. They all have a cytoskeleton, that's pretty easy to remember. Cyto, right? Cell, skeleton, structure. That's the internal framework. And then a cell membrane, the outer boundary, also known as the plasma membrane. So getting started first, prokaryotes, they were the first cells that came about. They're pretty simple. These are our bacteria cells. But what I want you to remember most about these guys is no nucleus. Actually, no membrane-bound organelles to begin with, but no nucleus. All of their DNA is just kind of floating out on the inside, and they're pretty simple and pretty primitive. A lot of these cells have cilia and flagella to help them move around, because a lot of these cells, or the whole organism, is just that one cell. So they're pretty simple, pretty basic. So these are our bacteria. These are actual images of bacteria from microscopes. See, they actually look this way, the whole capsule and the cilia and flagella. So you should be able to pause the video and make sure that you can find all of these things on this bacteria. If you cannot, refer back to your cell coloring that we did last week. So that's pretty much it for prokaryotes. They're pretty simple, pretty primitive. The ones that we're going to spend a bit more time on are the eukaryotic cells. These are the cells that are found in plants, animals, protists, and fungi. So these are all our upper level of organization cells. These are what these guys are going to be made out of. These guys have four main parts: the cell membrane that contains the cell, the cytoplasm, the nucleus. Eukaryotic cells have a nucleus, so that nucleus is where the DNA is contained. Also, eukaryotic cells have organelles, small structures that carry out functions. They're also, you can think of them as the little organs of the cell. The nucleus, what I need you to know about the nucleus is that it has a nuclear membrane which keeps everything inside of the nucleus, and nuclear pores. It contains all the cell's DNA in two forms. It has chromatin is how you normally find it. That's kind of like, it's all squiggly, all kind of a mess in the nucleus of that cell. And then it shrinks and all comes together and forms chromosomes during cell division. That just keeps DNA a little bit more organized, so that way it can divide evenly when that cell does it too. You should also know that there is something called the nucleolus, which is found on the inside of that cell, and that makes the cell's ribosomes. Moving on, we all know about the mitochondria, energy center of the cell. What I need you to know is that it turns food or sugar into chemical energy, also known as ATP. And we all know that mitochondria is the powerhouse of the cell, and that's what this guy looks like. Look for those inner squiggles in that mitochondria. Oops, sorry about that. So moving on to the Golgi apparatus. This is like, think of it as like a factory. It processes, packages, and exports, which means it gets it where it needs to go, proteins. So you can compare it to a factory or a post office because it packages all those proteins, but it also tells them where they need to go because these vesicles are formed and it transports those substances outside of the cell. Lysosomes are super important. They contain digestive enzymes. They break things down. You can think of them as a suicide sac. What I want you to think of is Tay-Sachs disease. It's when a person has defective lysosomes, and it's because they cannot break down those waste products. The substances kind of just build up on those toxic substances, build up and cause brain damage. So that's a disease of the lysosomes. When those lysosomes aren't breaking down things they're supposed to, that's when trouble comes about. Endoplasmic reticulum, also known as the ER, that's a transport system. Think of it as the highway, right? The rough ER contains ribosomes and the smooth ER does not, but they both are a transport system. Cytoskeletons, these guys right here, shape and movement for this one. I don't really care too much that you know microtubules versus microfilaments. But this is one thing that was not mentioned in our cell mobile project, so make sure you write this down in your chart, that centrioles. They are only in animal cells, guys, and they're used during cell division, and they look just like this, and they help the cell carry out cell division. So make sure you add those to your chart as well. And just know that cilia and flagella, they help the cell move. I'm not too worried that you know which way they go. And the vacuole is a storage area for water and other substances, and plants usually have a big one, this guy right here. Animal cells have a couple small ones. So they both have vacuoles, but they look different depending on what cell it is. One also important thing to remember about cells is it's all about proteins. Cells are like little factories, that little protein making factories. The DNA has the instructions to build proteins. Those instructions are then sent to the ribosomes. The ribosomes build the proteins and sends it down that through the ER, that transportation highway. And the proteins go to the Golgi, which they are then packaged and shipped to other parts of the cell or other parts of the body. That's protein. You should know the steps of protein synthesis right now at this level. Make sure you can label everything on this cell that you see here. Make sure you can label its structure, but that you also know the function of the animal cell and of that's actually what a real animal cell looks like. Those are cheek cells. The nucleus is right in there, and the cell membrane's out through here. So now, guys, I want you to pause this video and make sure that you can label all of the organelles that were on your chart. You should be able to label those on this cell right here. And remember, this is the animal cell. Moving on to plant cells now. The main difference is plant cells have a cell wall, chloroplast, and that central vacuole instead of those small vacuoles around the cell. So the cell wall provides additional support. It's like it goes outside the cell membrane and keeps it kind of hard. Chloroplasts, they have that green pigment and they do photosynthesis, and that big central vacuole. So can you name the parts of this plant cell? Pause the video, see if you can. This is what actual plant cells look like. And by now you should be able to see the animal versus the plant cell. I just want to briefly mention that mitochondria and chloroplasts, they have their own DNA, which means it's going to be similar to the DNA found in the nucleus, but they actually do have their own unique DNA. There is a theory called the endosymbiosis theory, which states that eukaryotic cells evolved when a prokaryotic cell engulfed another cell, and from there they had a symbiotic relationship, which they benefited off of one another, and that's how we got our mitochondria and our chloroplasts. Remember that our cell membrane is what we call selectively permeable, which means it regulates what comes into and out of the cell. It selects what can permeate it. It's composed of a double layer of phospholipids, which are these guys right here. Inside is hydrophobic. It does not like water. Outside is hydrophilic, so it can go with the water. And you have those transmembrane proteins all the way throughout. All right, guys, and that's it. If you were able to follow everything and knew what I was talking about, you will be good to go for this unit. Have a good day.